Good afternoon, everybody. Good afternoon. Can you all hear me? And can you all hear me properly, everybody? Yes. So today we will be dealing with the next lecture of biomolecules, that is the nucleic acid. Okay, let me share the screen for you and let's move with today's super amazing class, everybody. Done? Am I properly and clearly audible and the screen clearly visible to each one of you? Yes, sir, no, sir, whatever. On the chat box, everybody. Yes. So today's topic, guys, is nucleic acid and DNA and RNA, the two common and familiar terms for everybody, I hope, right? Nucleic acid, you are all aware about. DNA, the two terms, deoxyribonucleic acid, DNA and RNA, right? And today we'll be discussing about these two components of biomolecules, that is DNA and RNA. Okay, everybody. Those who are watching the session right now, don't forget to hit the like button, do share it with your friends and subscribe the amazing platform for more updates on biology. Guys, don't forget today sharp at 9 p.m. you will be having NCERT line by line quiz, line by line discussion of some of the important questions of biomolecules. Okay, so don't forget to attend it because we will be doing very important NCERT direct question. Okay, everybody. Yes, this is our Telegram group at t.me/NEET_biopoint where daily session updates, regular discussion, polls and quizzes. Please do link us directly given in the description box. Those who are watching this session, if you have not become a part of our small family of both a NEET 2023, NEET 2021, NEET 2022 of BioPoint, don't forget to click the link, direct join our Telegram group. Ready, everybody? Just tell me yes or no on the chat box. Is everybody ready? Yes? Nobody is there? Actually, I cannot receive any of your messages. What happened? Let me anyway refresh. 1 second. Yes, ready everybody? Just tell me. Yes, Anantu. Very good. So guys, today we are going to discuss about nitrogen bases. First of all, guys, before we start, I have to tell you something important, that is, listen very carefully. Guys, there are two types of nucleic acid. What are the two different types of nucleic acid? That is DNA and RNA. What are the two types? It is DNA and RNA. Guys, from the diagram itself, it's absolutely clear for us that is DNA is the double helical in nature. Okay, DNA will be like this. And what is the RNA will be like a single stranded. So DNA are double stranded molecule, whereas RNA is single stranded. Okay, point number one, that makes two different types of nucleic acid. And you have to make sure and you have to understand that DNA, what is the full form of DNA? Deoxyribonucleic acid, deoxyribonucleic acid. Okay, the name is actually, it's very simple, that is deoxyribose. Deoxyribonucleic acid. I will tell you, everybody, listen carefully. Deoxyribose. So the sugar which is present inside the DNA, the name is deoxyribose. Whereas in case of RNA, it is ribonucleic acid. Okay, RNA, it is ribonucleic acid. So ribose is the sugar present inside the RNA. Okay, the nucleic acid containing ribose sugar and the nucleic acid containing deoxyribose sugar. Done? Guys, this making unit or the building blocks of DNA, you will call them as nucleotides. Okay, everybody, the building units of DNA is known as nucleotides. Okay, how is nucleotides formed? A group of nucleosides. I will tell you what is this nucleotides, nucleosides and all. Nucleosides plus a phosphate group attached together to form a nucleotide. Okay, everybody. What is this nucleoside? Nucleoside is actually formed from a sugar and a pentose sugar plus a nitrogen base. So guys, nucleotide is equal to nucleotides plus phosphate. Done. Next one is nucleosides. Nucleoside is sugar plus nitrogen base. Clear, everybody? Is it clear? Guys, in a DNA or an RNA, in a nucleic acid general, I am telling, in a nucleic acid generally, I am telling you, that is, it has three main components. How many components? It has three main components. The first one is a nitrogen base. There is differences in each of them. That is what makes a difference. Okay, everybody. So a nitrogen base. Second one is a pentose sugar. Guys, what is pent? Pent means 5 carbon. 5 carbon sugar. Okay. And the third one is a phosphate group. Done, everybody. Phosphate group. Clear? Clear, everybody? Please do respond to me in the chat box. So these are the three main components. These are the three main components which you can see in a nucleic acid. That is nitrogen base, pentose sugar and the phosphate groups are the three main components that you will see in a nucleic acid. Done, everybody? Yes. Anyway, let's move on. Guys, nitrogen bases can be mainly classified into two. Okay, nitrogen bases can be mainly classified into two based upon the number of cyclic rings present in their structure. Based upon what? Based upon the number of cyclic ring present in their structure. That is, those structures which are having a double ring, you will call them as purines. And those structures which are having a single ring, you will call them as pyrimidine. So what are it? Purines and pyrimidines are the two different components. Done, everybody? Clear, everybody? Absolutely perfecto. Guys, purines means they will be having two benzene ring or they will be double ring compound. Whereas pyrimidines, they will be having only a single ring. And one more thing I have to tell you over here. Purines can be of two types. Okay, there are two things that come under this purine. That is adenine represented by the letter A, guanine which is represented by the letter G. Okay, whereas pyrimidines again can be classified into three, that is cytosine, oh my god, cytosine, uracil. Uracil is seen only in case of RNA. And one more thing, that is the thiamine. The thiamine is actually seen in DNA. So sometimes a question can come. Those who are having a blurred screen, please, there is 3 dots on the top or bottom of your screen. Please do click it, change the video quality so that you will be able to see the screen absolutely clear. Okay. Name the pyrimidine base which is seen only in RNA. The answer is uracil. Name the pyrimidine base which is seen only in DNA. It is thiamine. Okay, rest everything is found in both DNA and RNA. Let's anyway move on to the session. Okay, everybody. So listen carefully. Nitrogen bases, they are nitrogen containing cyclic compounds which are present in nucleic acid. Look, can you see a double ring compound? Guys, can you see a double ring compound? Oh my god. So guys, this is a double ring compound. So what is it? It is a purine. It is an adenine. And look over here. Here you will find a single ring. It is a pyrimidine. It is a uracil shown over here. So yes, Binaya, good afternoon. I hope you might have not watched yesterday's lecture because I have not found your comment for the homework. Yes, only Amni have commented me after that. Yes, everybody. Okay, done. So nitrogen bases are nitrogen containing cyclic compounds which are found in the nucleic acid. They are having 2 different types, that is purines and pyrimidines. Purines comprises of adenine and guanine. Pyrimidines comprise of cytosine, thiamine and uracil. Okay, everybody, clear? These structures are important. Guys, just you have to have a look over here, but because when you ask or when you attend a question, you will see these diagrams given and they'll be asking you to identify whether it is a nitrogen base, is it a nucleoside or is it a nucleotide. We'll be learning all those things. So learn along with me, everybody. Purines and pyrimidines. Purines, adenine, guanine, and pyrimidines is, purines are adenine and guanine, pyrimidine, cytosine, thiamine, and uracil. Is it clear, everybody? Guys, when this nitrogen base is linked with a sugar by a bond, we will learn about what is that bond in the upcoming slide. Okay. Yes. Guys, when a nitrogen base and a sugar are linked together, that structure you will learn as a nucleoside. Guys, this is a very important diagram, very important area from where repeated question come for your NEET examination. Okay. Yes, everybody. So nitrogen base combines with a sugar, you will call them as the nucleoside. Okay. Nitrogen base plus sugar gives a nucleoside. Adenine, oh my god. Adenine and guanine, adenosine is the name. Guanine plus sugar, then guanosine. Cytosine plus sugar, cytidine. Thiamine plus sugar, thymidine. Uracil plus sugar, uridine. Learn each of these terms. Okay, adenine, guanine, cytosine, thiamine, uracil. So what is the term of the respective nucleosides formed? What is the term for the respective nucleotides formed? It is adenosine, it is guanosine, it is cytidine, thymidine, and uridine. Guys, in your textbook, you have the diagram of adenosine and uridine in your NCERT textbook. So it is actually representing the nucleoside. Are you getting my point? Are you getting my point, everybody? Are you all getting the points very clearly? Let me ask you first of all. So guys, what were the nitrogen bases? Adenine, guanine, cytosine, uracil, and thiamine. And what are the nucleotides? And what are the nucleosides mentioned? It is Adenosine, guanosine, cytidine, thymidine, and uridine. Guys, are you understanding me? Only Anandu is there in the class. Oh my god, what is this? Binay and Yamini, are you keeping quiet since you are not understanding, or you are not there in the class? What is actually the conclusion that I should derive? If you need me to explain anything, I am ready to explain. Please do tell me. Okay, everybody. Okay, Binaya. So nucleosides means it is a combination of a nitrogen base and a sugar. So in your textbook, you have been given two different diagrams, that is adenosine and uridine. So you have to be very much clear about these two diagrams. Okay, let's move with the next one. Next is the nucleotides. Okay, everybody, next is the nucleotides. In the initial beginning itself, I have told you what is a nucleotide. Nucleotide is actually a combination of a nitrogen base plus a pentose sugar plus a phosphate group. Guys, have you anybody known about your results of your school, 12th standard and all? Have you anybody looked for that? How is it? What is the highest percentage in your respective schools? Yeah, within just one day gap, day after tomorrow, I am going to receive my dear children's results as well. Right? Maybe some of you at least may be having butterflies in your stomach. You'll be having, right? How many of you are super excited for your results? Yes, I think it's an official information which have been announced. Like CBSE 10th results would be announced soon on August 1st itself. So a fresh month with a fresh start, everything. It's time for you to gear up with your NEET preparation. The end of your CBSE class 10th is going to happen by your August 1st. I am waiting for everybody's results. Okay, I'm waiting for everybody's super amazing results, and I hope my fame of wall will be filled with each one of your photographs. Yes, everybody. Yes. So listen very carefully. I want the same results have to be in JEE 2023 and NEET 2023 as well. Okay, everybody. So nucleotides are actually formed by the combination of a nitrogen base, a sugar, and a phosphate group. So guys, adenine plus the nucleoside, that is adenosine, you will be getting the formation of an adenylate acid. In your textbook, only a single nucleotide sequence have been given, that is adenylic acid. That is an important one. Okay. So exactly like that, you have to study the name adenylic acid, guanylic acid, cytidilic acid, thymidilic acid, and uridylic acid. They are all different terms for what? Nucleotides. So guys, in your NEET exam, sometimes they have asked the direct question as well. Thymidilic acid is an example for option A nucleic acid, option B DNA, option C nucleoside, option D nucleotide. So your answer should be absolutely very simple. It is nucleotides. Done, and you must very much you have to be careful about it. Look, guys, adenine is a nitrogen base. Okay, adenine is a nitrogen base. This pentose carbon, okay, 5 carbon arranged, this is the sugar, pentose sugar, and this is the phosphate group. So what is actually the combination of these three known as? Comment me on the chat box, everybody. Combination of a nitrogen base, sugar, and a pentose sugar. Together you will call them as what? Yes, everybody, it is a nucleo Am I clearly audible? And look at the screen, everybody. Is the screen clearly visible to each one of you in the class? Yes? Very good. Yes? Yes, everybody? Yes, so listen carefully. Nitrogen-based nucleotides. Nucleotides are heterocyclic compounds. Okay, they are heterocyclic compound. Look at the wonderful image of DNA shown over here. So it is a double helical structure, right? It is a double helical structure. Okay, and the nucleic acid, DNA, and RNA are made up of nucleotides. Okay, everybody. Oh, you are getting the sounds of the temple. You may be getting disturbed. With that, yes, let me adjust the mic so that I could reduce the volume. Yes. Yes, listen very carefully. Okay. And guys, Binay and Yamni, are you all from your CBSE board or the state board? Just tell me on the chat box fast. Okay. Okay. So nucleotides are heterocyclic compounds and the nucleic acids, DNA and RNA are made up of simple units known as nucleotides. And what is a nucleotide? It is a combination of a phosphate, nitrogen base and what? Nitrogen base along with a sugar. Okay, so you are having a term 1 exam and this is biomolecules is gonna be our last chapter for term 1. So what is your plan is like should biopoint do with the revisions of the already completed chapters of term 1 or by the month of October only we need to revise it again? We should complete the portions of NEET examinations and or we should come back to the revision. What is your opinion? I just want to ask your opinion because biomolecules will be having 2 to 3 classes. Transport in plants will be having only 1 to 2 classes left out. What's your opinion? Should we move on with the NEET examination and by the month of September, October, we would come across. Your exam is November, right? CBSE term 1 exam. So should we start the revision of the previous chapters by the month of September, October, or should we again revise? Go back and revise and come back. Please just comment down. Just comment down the detailed way. Okay, I cannot understand to which are you agreeing. Should you need revision or we should concentrate on NEET till September? Okay, everybody. So guys, nucleic acid are the heteropolymer of nucleotides and they are linked together to form a polynucleotide chain. Okay, everybody. So I hope everybody will be going like that. Okay. Yes, guys, this is an absolute example for a nucleotide. Okay, a nitrogen base will be there, a sugar will be there, and a phosphate group will be there. So that is a nucleotide structure. Guys, a group of nucleotides will be joining together to form a polynucleotide chain. Okay, so this is the structure of a nucleotide. A group of nucleotide would be joining together to form a polynucleotide chain. So that is all about this. So this we have already discussed. I am going to explain or revise it again for you. Nucleic acid are of two types, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. And guys, in the last class also I told you about the same thing, that is DNA will show the secondary structure and it was proposed by Watson and Crick. So guys, we know that DNA have a double helical structure. DNA have a double helical structure and that double helical structure was proposed by Watson and Crick. Okay, those double helical structure was proposed by Watson and Crick. Done, everybody? Yes, so that's important. And guys, this structure which we will see, okay, this structure which we will be seeing, this is actually a secondary structure, just as how we learned for the protein. This is a secondary structure. Okay, so there are more than a dozen of DNA such as aDNA, bDNA, cDNA, ddNA, eDNA, zDNA and all. But the general DNA that we are concentrating while we will be studying and all those that we are studying are the structures of bDNA. Okay, bDNA is easy to isolate and easy to study. Okay, so DNA consists of two polynucleotide chain arranged anti-parallelly as a double helical structure. Okay, everybody. So I will be showing you one thing. Okay. Yes, perfecto. So guys, this is particular chain you suppose. Guys, this is one particular chain having a polarity. This polarity and all, what is this polarity and all, I am not going to explain right now. About this polarity and all, you will study in the next chapter, that is next year chapter. That is a molecular basis you will study. So this is having a polarity of 5-3-. And you are having another DNA also, another strand of DNA having an opposite polarity. So since they are having opposite polarity, you will be calling them as anti-parallel. What you will be calling them? You will be calling them as anti-parallel. Clear, everybody? So these two will get intertwined together. That is, this will get twisted. Okay, once again I'll draw that. Listen very carefully. Yes, this will get twisted like this. That is actually they'll twist in the right-handed fashion. Okay, they'll twist in a right-handed fashion. So this is having a polarity 5-3- and this is having a polarity of 3-5-. And they will be interconnected with the help of a hydrogen bond. Okay, they will be held together with the help of hydrogen bonding. Clear? That is what is mentioned over here. Okay guys, the secondary structure DNA will be consisting of a nitrogen base, deoxyribose sugar and a phosphate group. Yes, the backbone of the DNA is formed by the sugar phosphate. Okay, so this is a previous question. The backbone of the DNA is formed by dash. The backbone of the DNA is formed by sugar and phosphate, whereas the nitrogen bases will be projected out, just like the steps in a staircase. Okay, just like the steps in a staircase, you will find the nitrogen base. They will be getting projected from this particular backbone. So the backbone, that's very easy and a simple question, but a difficult thing to remember for everybody. You'll get easily forget those things. So backbone of a DNA is formed by the sugar phosphate bond, whereas steps are formed by the nitrogen bases, just like the steps in a staircase. So I have already told you that RNA and DNA are two different things. DNA is a double helical structure, RNA is a single stranded one. Okay, DNA, the another difference which I told you is that DNA have a nitrogen base of pyrimidine, thymidine, that is thymine, and RNA have uracil. Okay, so what are the four nitrogen bases present in DNA nucleic acid? Adenine, guanine, cytosine, thymine. Whereas in RNA it is adenine, guanine, cytosine, uracil. Okay, guys, adenine pairs with thymine. Adenine pairs with thymine. Adenine pairs with thymine having a 2 hydrogen bond. Okay, by 2 hydrogen bonds. Just exactly like that. Guanine pairs with cytosine with the help of 3 hydrogen bond. Just like this, guys. This is a This is what phosphate group. This is what I will enlarge the image for you so that you will be getting to know that very easily, everybody. Yes. So, guys, this is a structure for, yes, perfecto. I think right now it will be perfecto. So, guys, this is what, listen very carefully. Okay, guys, this is what it is, an example, that is phosphate. This is sugar. This is a nitrogen base, adenine. So adenine always pairs with thymine. There is no exception towards that. Adenine always pairs with thymine using 2 double bond. Always with 2 double bond, adenine and thymine. Okay, and guanine pairs with cytosine with the help of 3 bonds, 3 hydrogen bond. Not 2 double bonds, a single double bond. That is 2 pairs of bond. Thymine again bonds with adenine with 2 bonds, 2 hydrogen bonds. Then cytosine with guanine with the help of 3 bonds. That is important. Is that clear for everybody? 2 hydrogen bonds are present between adenine and thymine. 3 hydrogen bonds are present between guanine and cytosine in case of DNA. Now a confusion is there in your mind whether what will be then in RNA. RNA there is no thymine. In case of thymine you have to use uracil. That's the difference. Adenine and uracil pairs by 2 hydrogen bonds. Guanine and cytosine pairs by 3 hydrogen bonds. And guys, this is very important. A phosphate molecule links to the 3' carbon of the sugar to the 5' of one nucleotide to the 5' sugar of the next nucleotide. Just you have to identify, that is third carbon. Third carbon. Okay, a phosphate molecule will be attaching. Look, a phosphate molecule will be attaching to the third carbon of the nucleotide. So that nucleotide, from that nucleotide, you will be getting the 5th carbon atom will be attaching to the next sugar also. That is phosphate group as well. Okay. And this slide is an absolute important slide. So guys, comment on the chat box. What is the bond that you will see in between 2 different nitrogen bases? It is the hydrogen bond. What is the bond that you will see between a phosphate group and a sugar? Phosphate and sugar, you will find an ester bond. Between 2 nucleotides, that is between 2 ester bonds, you will find what? Phosphodiester bond. Between 2 nucleotide sequences, you will find phosphodiester bond. 3 bonds we are studying. Absolutely, it's very important. That is between nitrogen bases, you will find hydrogen bond. Between 2 phosphates, between a phosphate and a sugar. Okay, very super amazing important, super important. Okay, ester bond between a phosphate and a sugar, and between 2 nucleotides it is the phosphodiester bond. The bond between a sugar and a nitrogen base, there is one more connection also, right? A phosphate, that is sugar, and a nitrogen base is the N-glycosidic bond. Where, in which compound have we studied the N-glycosidic bond? In carbohydrate. In protein we have studied about the peptide bond. Yes, everybody? Yes. So guys, this you have to by heart. This you have to simply by heart. We'll study the details of this in your class 12th itself. One full turn of 10 steps contain 10 base pair. So guys, this one twist will contain 10 base pair. Length of one full turn will be 34 Armstrong. Okay, 34 Armstrong. At each step, the strand turns 36 degree. You have to study one full turn of DNA. BDNA will have 10 base pairs. And one length of full turn will be having 34 Armstrong. The length of one full turn is 34 Armstrong. Clear? Yes, I'm going to highlight the important points for everybody. Yes, living organisms have a number of carbon compounds in the heterocyclic rings are found. Some of them are nitrogen bases. That is adenine, guanine, cytosine, uracil and thymine. Guys, for seeing the slides very perfect and clearly, please change the video quality so that you will be able to see this. When found attached to sugar, when the nitrogen bases, okay, when the nitrogen bases are attached to a sugar, when this nitrogen bases are attached to a sugar, you will call them as what? You will call them as a nucleoside. And when a phosphate group is again added to this nucleoside, you will call them as nucleotides. And they will get esterified. That is, which bond is present between a nucleoside and a phosphate? It is the ester bond. Is that clear? And guys, what are the different names that you will give for the nucleosides? It is adenosine, it is guanosine, it is thymidine, uridine, and cytidine. So adenosine, guanosine, thymidine, uridine, and cytidine are the nucleosides. Whereas adenylate acid, thymidylate acid, guanylic acid, uridylic acid, and cytidylate acid are the nucleotides. Okay, and DNA and RNA functions as the basic genetic material. Clear? So this you have to be very careful. So guys, this is what I told you given in the NCERT textbook. Look, adenine. It is given in the NCERT textbook. Adenine, uracil, which is a pyrimidine, they are nitrogen bases. Okay, adenosine and uridine, they are nucleosides. And adenylic acid, it is a nucleotide given in the NCERT. This you can easily identify since they are having a double. Okay, since they are having a double ring. Since this compound is having a double ring. Okay, can you see double rings are there? So it comes under the adenine component. And since this compound is having a single ring, it comes under the pyrimidine. Okay, purines and pyrimidines. Clear? Don't get confused. I know it is a bit confusing, but it's anyway we have to study. Right? So, the other type of macromolecule that we would find in the acid insoluble fraction, okay, this is in the acid insoluble fraction, is the nucleic acid. These are polynucleotides. Okay, I'll reduce the size so that I can mark it. Okay, guys, acid insoluble fraction, it is a nucleic acid. They are polynucleotides. Clear? The nucleic acid, the building block of nucleic acid is nucleotide. Vinaya and Yamini, Anandu, are you all getting the points? Yes? So you'll have a heterocyclic compound, that is a nitrogen base. Second is a monosaccharide, that is a pentose sugar. And third is a phosphoric acid or a phosphate molecule. So a combination formed by all these three, you will call them as what? A nucleotide. So what are the different types of nitrogen bases? Adenine, guanine, uracil, cytosine, and thymine. Yes, everybody? Yes. So adenine and guanine are substituted. Adenine and guanine are substituted purines. Adenine and guanine are substituted purines. Adenine and guanine are substituted purines, while the rest are substituted pyrimidines. Okay. The skeletal heterocyclic ring is called purine and pyrimidine. The sugar is a ribose sugar or 2-deoxyribose sugar. Okay, there are two nucleic acid, deoxyribonucleic acid, which is a DNA that contains deoxyribose sugar. Once again, I'll highlight deoxyribose sugar, deoxyribonucleic acid, and ribose sugar in ribonucleic acid. That's very important. Clear, everybody? Clear, everybody? Is that absolutely clear for everybody? And guys, you have to learn that in proteins, that is, amino acids are linked by a peptide bond. Amino acids are linked by a peptide bond. Carbohydrates or polysaccharides are also linked by a glycosidic bond. Okay, so the bond which is present between the phosphate group and the sugar, you will call them as a ester bond. The bond which is formed between a sugar and a nitrogen base is the N-glycosidic bond. N-glycosidic bond connects nitrogen base and the sugar. Bond present between two nucleotides is phosphodiester bond. Okay, 4 bonds you have to study. Yes, and we already studied nucleic acid can be DNA, can exist in many forms, but the common DNA that we use for our studies is the bDNA. The bDNA exhibits a double helical structure, and that double helical structure of DNA was proposed by Watson and Crick. Okay, and these contains 2 polynucleotide chains that are running anti-parallel to each other. And the backbone of the bDNA is formed by the sugar and the phosphate, whereas the nitrogen bases are projected from the surface, just like the steps in a stair. Nitrogen bases such as adenine and thymine in case of DNA, or adenine and uracil in case of RNA, are held together with the help of 2 hydrogen bonds. Whereas you will see that the bond formed between cytosine, guanosine, and cytosine, guanosine or guanosine cytosine, that will be what? That will be having what? It will be having 3 hydrogen bonds in between. Done, everybody. This is a diagram of a bDNA which is seen in your NCERT textbook. Look, 2 hydrogen bonds are formed over here. Yes, here you will find 2 hydrogen bonds, 2 hydrogen bonds, and but here you will find guanosine and cytosine, you will find 3 hydrogen bonds. Done, everybody. And this was also what I have told you, that is hydrogen bond will be 3 hydrogen bond between A and T, and G and C there will be 3 hydrogen bonds. Okay, will appear like a helical staircase, and you will have the strand turns at an angle of 36 degrees. One full turn of a helical strand would involve 10 steps or 10 base pair. The pitch, I will tell you in that class 12th, I'll tell you in detail about what is this pitch and all. So pitch of the DNA will be 34 Armstrong. Okay, 34 Armstrong. And in higher classes, you will be told that there are more than a dozen of DNA named after British alphabet with a unique structural feature. So guys, what is the DNA that we will be discussing? It is the bDNA. Clear? Yes, up to here is the portions clear for everybody. So thank you, everybody. Sharp at 7 p.m. you will be having the lecture on transport in plants, and at 9 you will be having the NCERT line by line quiz on biomolecules. So yesterday I have posted a photo in our group regarding, and I have also indicated an underline over there in the textbook regarding like you have to learn up to there. Okay, I have told you. Okay, so tomorrow, that is at 9 p.m. come prepared with the topic by 4 o'clock and all. Be prepared. Please prepare the topic for the 9 p.m. session so that you will get a detailed way. You will be able to solve the questions as fast as possible. And when you are coming to attend the session at 9 p.m., make sure you take an error copy with you, a full scan paper or whatever. Okay, you can take a sheet of paper, any sheet of paper, or the backside of your notebook itself, or a simple sheet of paper, you can take it, and you have to write down. First of all, you have to write down the answers on your notebooks or your paper sheets or your error copies or whatever you are taking, and you have to comment down the answers. And finally, when the session is over, you have to comment to me on the Telegram as well as on the chat box, in comment box, especially in the comment box, and personally to me in Telegram. You have to tell me how many marks have you scored out of the 20 questions we will be doing. So 20 questions you will be doing in today's session. So out of 20, how many marks are you getting? You have to comment down. Bye bye, everybody. Stay tuned for the 7 p.m. transport in plants lecture number 5.